So my talk today is called Metabolic and Neuroregulation of Immunity and Cancer. This is a painting by Wang Ying from Shanghai, who's a very good friend. And I guess the message of the painting is that we have to dance together in order to have fun. Now, about 2,500 years ago, 500 kilometers south of here, was someone who was born. His name was Confucius. And he said, we have two lives. The second one begins when we realize we only have one. I think this is very profound for those of us who have had traumatic experiences with our loved ones or ourselves. For the audience, medical scientists, you also have two lives. And the second one begins when you realize immunology is the orchestra of most life symphonies. So that is very important. I'm only going to concentrate on cancer, as I have no time to discuss other properties. Now, central to the immune system are many receptors, one of which is the T-cell receptor. It is the receptor that differentiates between self and non-self. And of course, for self, you should not attack, and for non-self, you should. So within this T-cell receptor interaction is this peptide and the HLA. And of course, today we know that cancer can be treated very effectively with CAR-T, and also TCR-T have also been approved for uveal melanoma for GP100 HLA-A0201. So from my first slide, I'm trying to illustrate to you, at the end of the day, our immune system, especially in where we're talking about cancer, is sitting in a place where the tumor microenvironment is affected by metabolic adaptation, by genomic instability, and that is perhaps the answer to whether we can be more or less successful with immunotherapy. Now, nothing is more kind of like poignant than the fact that a very important and crucial isocitrate dehydrogenase metabolic enzyme is actually mutated in multiple cancers. It was first discovered by Bert Vogelstein in gliomas. Our laboratory, together with a French laboratory, Philippe Galad, discovered that T-cell lymphomas also have mutations of isocitrate dehydrogenase. As well, of course, AML discovered by groups in Washington University, Tim Lay's. Now, we, of course, just really like the idea of having mouse tell us what to do. And so a Japanese postdoc and a German postdoc worked very hard to try to understand why this mutation would cause cancer. It turned out it was not a gain of function. It was not a loss of function. It was a neomorphic mutation. What it really means is that the mutant take the product of the wild type into a brand new metabolite called 2-hydroxyglutarate. And what this oncometabolite does, it blocks the function of alpha-ketoglutarate, which is involved in 70 different alpha-ketoglutarate-dependent dehydrogenases. And in so doing, it affects not only metabolism, redox hypoxia, epigenetics, and other kind of enzymes and metabolic changes that requires alpha-ketoglutarate. More recently, we found something quite interesting, was that the mutation found in the T-cell lymphoma completely changes the tumor microenvironment. What it does is that it upregulates ICOS PD-1, but it downregulates FAST ligand in the T cells. That results in an upregulation of the co-stimulatory molecules, but no death. And in such so the spleen weight goes up, and eventually you get cancer. Now, totally unexpectedly, but very gratitude-wise, that the drugs that we made together with Craig Thompson and Luke Kentley through the company Agios actually has benefit to acute myeloblastic leukemia. It's also been approved for cholangiocarcinoma, and glioma, of course, is something that we are all very concerned. These are the trials, and it shows that the company Servier, which is in France, has just finished the phase 3 clinical trial on an inhibitor that penetrates into the brain. And gratitude,ly it has now met its primary endpoint, and hopefully there will be further progress along this way. So there we are. We found metabolic adaptation being part of the kind of immune modulation, but also that metabolic adaptation can also affect genomic instability. So a Japanese postdoc and Taiwanese and I also show that the mutation of isocitrate dehydrogenase actually also reduce the level of ATM, which is, of course, a very important enzyme in DNA repair. And in so doing, there is a reduction in DNA repair. So that brings us to the idea that we could actually affect DNA repair, and it's part of the development of the cancer. But can we actually take advantage of that to treat cancers? And of course, we know that PARP inhibitors and topoisomerase inhibitors are effective and been approved to treat cancers that are genomically unstable. So about 15 years ago, we pursued the same idea. And the idea was, can we find other enzymes that are involved in contributing to genomic instability? And that genomic instability could be harvested in some ways to actually help in treating the patients. So one of the enzymes was proteokinase 4, which is involved in central duplication. And the other enzyme is TTK or MPS 1. As you can see, these enzymes are upregulated 10 to 100-fold in these different kinds of cancers. So together, our hospital in Toronto spun out a company called Treadwell, and they have now developed 2 drugs, both of which are now in phase 2. The first one, as you can see here, this is a RAS-mutated colorectal cancer in late stages. And this is the second drug, which you can see here. Even in monotherapy, we are seeing some refractory relapse breast cancer responses. Now, why am I showing you this? I'm showing you this is because genomic instability also contributes to immunotherapy. Several groups have collaborated with us or independent of us, utilized our compounds. You can see here one at Harvard and others in Hong Kong, have been able to show that these compounds were able to trigger some kind of an immune response by perhaps contributing to the immunosurveillance. And of course, we are lucky to show that this paper that just appeared 2 weeks ago, we actually came across that these compounds are able to activate this CGAS STING pathway. And of course, the next speaker will be actually James Chan, credited by all of us for discovering this very important enzyme. For the rest of the time, I'm going to go back to something that perhaps not all of you have been thinking about. This is Richard Feynman, and he said, I would rather have questions that can't be answered than answers that can't be questioned. Now here are some of the slides that have been shown to me over and over again by pathologists. One of the curious findings is that the tumors appear to have a wall that prevent the T cells from going into the tumors to attack the tumors. And then what would be this wall? And for those of you who are familiar with Helen of Troy, in other words, what are the Trojan horses that would allow us to manipulate the system to attack the tumors with the T cells? Now go back to someone that while I was training in Toronto that I have met. His name is Emmanuel Faber. In the 1980s and the 1990s, he and the Japanese group were able to show that if you leave choline out of the diet of rats, they will all come down with liver cancer. So what is choline? Choline, of course, is something that is very prominent on our minds. It is present in a lot of good food and a lot of bad food. Choline has three major pathways. The first is that together with methionine, it is involved in epigenetic changes. The second is that choline is part of the precursor to the synthesis of phosphatidylcholine and also sphingomyelin. But the last thing you would ever think of is that choline, together with acetyl-CoA, makes the neurotransmitter acetylcholine. So that brings me to my last 5 minutes or so, or whatever time that I have. Does the brain talk to our immune system? Of course, this is not a unique idea, but someone has to prove it. Okay. So back 170 years ago, the Germans had been stimulating the nerves that go into the spleen. Rudolf Wagner did it with dogs. Jakob Henle did it in a decapitated human, that when they stimulated the nerves, the spleen moved. Well, what does that mean? That means perhaps the brain talks to the immune system, as spleen is mostly, if not entirely, immune-related. So let's go back to a very simple paradigm. Parasympathetic nerves makes acetylcholine and rest and digest. Sympathetic nerves make norepinephrine, and it is fight and flight. So when the Germans were stimulating the vagal nerve that goes to the spleen moved, you expect acetylcholine. Everything went well. 100 years ago, Dale and Dudley found acetylcholine in the spleen. 1960, Brandon and Rand stimulated the nerves, and they found more acetylcholine. So everything was fine, except nobody can find parasympathetic nerves in the spleen. The only thing they find is sympathetic nerves, which is stained by tyrosine hydroxylase, and they make norepinephrine. So perhaps the vagal nerve that goes to the spleen just before it goes into the spleen, it hits the celiac ganglion and switch completely opposite direction to make norepinephrine. Then who is making the acetylcholine? Who are these cells? So I was lucky to have met a brilliant neurosurgeon in New York by the name of Kevin Tracy. And starting from 2008, we started collaborating. And what we did was very simple. We made a mouse in which you have chat GFP. This is a green fluorescent protein, and don't confuse this with chat GPT. Now, these green fluorescent protein told us that T cells in the first paper and B cells in the first paper make acetylcholine. In fact, you can stimulate the vagal nerve and they make acetylcholine, or you can add norepinephrine and they will make acetylcholine. So Maureen Cox, an American postdoc, came to the lab. And what she did was to take a mouse which has a background of about 4%, 2% acetylcholine that are CD4 cells. CD8 cells make nothing at all. So after 8 days of infection with a virus, she discovered there was a tremendous, enormous amount of acetylcholine made by both CD4 and CD8 T cells. In fact, in some mice, the number of CD4 and CD8 T cells exceeds 70, 80%. So, but this doesn't really prove anything. You have to make a mouse in which the acetylcholine transferase is deleted in CD4, CD8 T cells. When that happens, you give the mouse a virus. And you see if whether it will, without acetylcholine transferase, clear the virus. And the answer was clearly not. That means acetylcholine transferase is needed by the T cells to clear the virus. Well, what is the mechanism? One of the mechanisms is that the T cells bring the acetylcholine directly to the endothelial cells and say, knock, 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 let me open the door, and I need to go in. So we are now facing a situation where I would very fast tell you what is going on. I think this is the tip of the iceberg. First of all, right in the audience, Kai Chi had done some brilliant work. His group found that if you stimulate the nerves into the spleen, what you see is that there is plasma cells and there are acetylcholine transferase that are made by T cells, which affects the germinal center antibody formation. In collaboration with Peter Olesen, we showed that the T cells making acetylcholine actually can reduce the blood pressure of the mice. This, he, without us, had just published a brilliant paper that showed the first time in human that severe circulatory failures is correlated with the number of acetylcholine positive CD4 T cells in the patients in a very dramatic way. Very quickly, we've also shown that in EAE, the Th17 cells, that if you take the Th17 cells, which are acetylcholine positive, they cause EAE. And if you take the acetylcholine negative Th17s, they don't do that. So my final story, and I'll try to do this in 2 minutes, is cancer. This is a liver, and these are sympathetic nerves. They make norepinephrine. But you can see here very clearly that the sympathetic nerves is completely wrapping around only the portal veins, as if they actually have something to do with either controlling or affecting it. So Chongqing Zhang made a model, and without the CD4 T cells making the acetylcholine, you can clearly see there's a dramatic increase in the number of nodules in the liver cancer, and the mouse died earlier. So all he also did was to very systematically categorize 11 subtypes of T cells with and without acetylcholine transferase. And what he's able to show is that the acetylcholine that is made within the liver is able to downregulate the Tregs and allow the T cells to actually work together with downregulating PD-1. In the absence of acetylcholine in the thymus, in the liver by the CD4 T cell, Tregs go up and PD-1 goes up, and that may explain why the liver has more nodules. What is even more stunning is that he was able to show that the T cells appear to be oligoclonal, and more importantly, the T cells that are oligoclonal are actually making acetylcholine. Now, this is, of course, just going to the data, and we were surprised, or perhaps not surprised, to find that in liver cancer patients, if they do express high level of the acetylcholine receptor CHOM3 and CHOM5, they live 500 to 1,000 days longer. So my final 2 slides is that I begged the clinicians downstairs, Eric Chen, and I say, Eric, please, can you measure the level of choline in the patients undergoing through immunotherapy? And they said, why? I said, I don't know why. Would you do it? And they did it, and here's the results. If the patient has high choline, the prognosis is much, much better. So are we totally out of our minds? I don't know. Maybe we are. Thank you very much.